Good afternoon, everybody, and it's a pleasure to welcome Yannis KOMINIS from the Department of Physics at the University of Crete. He earned his PhD in 2001 from Princeton University, working, let's say, general topics of quantum optics, let's say, umbrella of quantum optics. Then he spent there another year as a postdoc, and then moved to 2004 in Berkeley laboratory,
2003 Berkeley, yeah.
Laboratories. And he is from 2018 associate professor at the Department of Physics, University of Crete, with original and pioneering work in quantum effects in biological systems, and that's today we will hear already talk about his efforts. Thank you very much.
Thank you. Thanks for the introduction. It's a great pleasure to be here. I'll try to squeeze in this talk work that took place over many years, like the last 10 years or so, but also towards the end report some more recent results. As Kaslav said, I'm at the University of Crete at the physics department, right in the middle of the Mediterranean. This is the campus, the University of Crete campus in Heraklion. Physics department is right there. Before getting into today's topic, I will just give you a couple of slides of what other stuff we're working on in the group. So we are doing also experiments, although today's talk is about is theoretical work, quantum biology work is theoretical. We're doing experiments in quantum sensing with hot atomic vapors. These are inside glass cells. We have like cesium vapors or rubidium vapors, and they are systems used for sensitive magnetometry, like being pumped with lasers and probed again with lasers. For the last few years, we have been studying spin noise, so spontaneous quantum fluctuations of the collective spin of the atoms sitting inside those cells. Another direction, which is again experimental, is what we call quantum vision. We study the visual system. We started with reporting a methodology to biometrically identify human beings based on their perception of very weak light, of like few photons. And after that, we started doing pupillometry, that is, measure the change in the pupil diameter upon illumination with laser light, hoping to understand the brain circuits that are responsible for pupil diameter changes, and thus use this for diagnostic purposes of various pathologies of the visual pathway and the nervous system. So now to quantum biology, as I said, our work started more than 10 years ago. It has been so far theoretical. At some point, I hope that we can do some experiments as well. In 2004, there was a workshop about whether quantum effects in biology are trivial or not. In fact, Anton Zeilinger was one of the participants, and he said that it would be extremely non-trivial if quantum mechanics did not play a role in living systems. It would be the only area in which we know quantum mechanics is not at work. Another quote by Weismann was that a better definition of what is non-trivial is not what will surprise physicists, but something that would convince biologists that they need to learn quantum physics. So, are we there yet? Well, let's see.
It's an accident that Howard Weismann is visiting us today.
Oh, yes? Okay. All right. So, of course, everything is quantum mechanical. When we're talking about structure of matter, even biological matter, you know, there are biomolecules like DNA, nucleotides. To understand the structure of such biomolecules, we solve Schrödinger's equation in some potential and hence understand electronic structure, all right? But this is not what we refer to when we talk about quantum biology. We mostly refer to situations like this. This is a picture from a book of Aharonov, where this skier shows the tracks of another skier, you know, so it looks as if this guy went through the tree. So it is about superpositions. So the heart of quantum, as you know, are fragile superpositions. For example, here I have a superposition state of a spin system, constant up plus another constant down, spin down. So the second quantum revolution relies on quantum coherence and entanglement. And these effects are rather fragile. That's why a main goal of this technology is to protect coherence and entanglement. And there are various technologies, like, of course, quantum computers, but also atomic clocks, quantum dots, atomic magnetometers that exhibit coherent effects. And they are the better and the more competitive, the better they protect coherence from the environment. Quantum measurement is also important in this discussion, and quantum measurement gives us information about the system we are studying. So we have a little quantum system that couples to some apparatus that might be another quantum system, and then there is a bigger apparatus, and then at some point we realize that the measurement pointer shows 5 volts and the spin is up. So in this process, we have also decoherence going on, and measurement gives us information, but also decoheres the system. And, you know, the interplay of quantum dynamics, coherent quantum dynamics, and decoherence is somewhat what defines quantum biology. So there is a map of various processes, and in the x-axis is the length scale of this process, and the y-axis is the time scale. This map is from a review on quantum biology by Huelga and Plenio. And for example, here at angstrom scale and femtosecond time scale, we have electronic dynamics. But here we can go up to the brain at you know, micrometer scale and millisecond time scale. And the question is, okay, there are a number of biological processes in this map, like brain, protein folding, et cetera, et cetera. Where is the barrier between quantum effects and classical world? So there must be some transition region beyond which we have quantum world dominate, and after which we have classical world, okay? So where is this quantum to classical transition, and what sort of biological effects are inside the quantum world? That's one basic question defining quantum biology. So another question that we can use to define what is quantum biology is where on the map of biological effects is the quantum classical transition, what I said before. The second question is what sort of quantum coherent effects do biological processes use to their advantage? That is, it's one thing to have a coherent effect, but the other thing is it any useful? That's another question. And a third aspect of quantum biology is what sort of quantum technology can we use to study biological processes, whether they are quantum or classical. So that's another point. I will not touch upon this, but it is being discussed lately. And of course, Schrödinger's house, it's not far from here, right? You can always wonder what is life and put this question in the general context of quantum physics. So that's another way to ask what is quantum biology. So a specific system that has attracted both my attention and the attention of a large community, both in quantum physics and in physical chemistry, and of course biology, is the Avian Compass. It has been known for a long time that avian species can orient in the magnetic field of the Earth. These are experiments typically performed in cages where people apply well-controlled magnetic fields. They can show that they monitor the tracks of the birds inside those cages, and look at their distribution in space. And then they rotate the magnetic field and see that the whole distribution is rotating also. So that shows that these birds can feel the magnetic field. And moreover, there have been recent experiments using radio frequency to irradiate those birds. And when the intensity of this radio frequency is high enough, the compass does not work anymore. So they get disoriented. So that points to the radical pair mechanism that I will discuss later. And it's pretty much settled that the Avian Compass is based on the radical pair mechanism. So what is this mechanism? It's a biochemical reaction. Where does it take place? Well, let's see. We start from Earth with its magnetic field. We go to the bird. We look at the bird's eye, the retina of the bird. There are proteins, cryptochromes, sitting on the retina of the bird, and inside there is a biochemical reaction. Now, that was proposed already in the late 60s by Klaus Schulten, a late German biophysicist. So the model has been there for a while and has been refined along the years. This is a nice paper by Ritz, Adam, and Schulten that discuss how this model works in detail. So what is this sort of biochemical reaction that at the end has to be magnetic sensitive, because the species can sense the magnetic field. So we need a chemical reaction that is field dependent. So before I discuss this reaction, I should mention that the same chemical reaction, called radical pair reaction, takes place in photosynthesis, and people study photosynthetic reaction dynamics using molecules inside an NMR magnet. So when there is light, photosynthesis needs light, they measure nice NMR spectra, and these spectra reflect the so-called kidnap effect and are also a manifestation of the same mechanism responsible for the compass of the avian species. All this discussion falls under the umbrella of spin chemistry, which is the field studying the effect of electronic and nuclear spins in chemical reactions. And I don't want to leave you with the impression that we're only talking about the Avian Compass, and which might seem a hypothetical model. There are specific experiments with molecules, like this is a synthesized molecule used by the Oxford group to get real experimental data manifesting magnetic field effects with chemical reactions. So a more recent paper doing pump probe spectroscopy and displaying quantum bits, again with this kind of molecules, was by the group of Lambert a couple of years ago. So there are laboratory experiments that discuss these kinds of reaction I will talk about. And of course, there is the interpretation of their use in the Avian Compass. So I just want to connect the macroscopic biological world with laboratory systems studied by physicists and chemists. So how does this mechanism work? The mechanism needs a photon. So there is a molecule, donor-acceptor couple, dyad, that absorbs a photon, is excited, and then one electron of the donor hops to the acceptor. So we have the creation of a radical ion at the donor and another radical ion at the acceptor. That's why we call this a radical ion pair, and the mechanism is the radical pair mechanism. So initially, the two unpaired spins sitting at the donor and the acceptor are in a singlet state. The singlet state is a spin zero state. It's like the state of the helium atom. Okay. And now, after the creation of this radical pair at time zero, because these dynamics are very fast, there are magnetic interactions within the molecules that result in a singlet triplet coherent oscillation. That is, the two electrons from the singlet state oscillate to the triplet and back. And while this is going on, the electron might at some point come back from the acceptor to the donor and result in the reaction product, which is a neutral molecule, either the original one through the singlet channel or another triplet molecule through the triplet channel. Okay? So the reaction starts from this guy, and it completes leading back to either this guy or this guy. These are the reaction products. Okay? In reality, the system is a little bit more complicated because here is an example of a donor molecule, and here is an acceptor molecule. So we have one electron spin in the donor, one electron spin in the acceptor, but each of those molecules has a number of nuclear spins. So the density matrix of the system is rather large in principle. It can have tens of nuclear spins because both donor and acceptor can have a number of nuclear spins. So you might end up with a large system. So we are talking about a spin system here. And when we refer to coherence, we refer to spin coherence. Okay? In practice, we simulate these systems by forgetting about too many nuclear spins and to keep only one or two nuclear spins so that our simulations are manageable because the dimension of the system blows up very quickly if you add qubits. So we have two electronic spins and a number of nuclear spins comprising our system. Now, why is the whole thing magnetic sensitive? Because the donor electronic spin couples to all nuclear spins of the donor through the hyperfine interaction. The same for the acceptor's electron spin. It couples to all the nuclear spins of the acceptor. So we have a number of hyperfine terms. But the donor electron spin and acceptor electron spin also couples to the external magnetic field through the Zeeman interaction. So the Hamiltonian driving this conversion has inside the Zeeman term, and therefore the magnetic field of the Earth, or the laboratory magnetic field, comes in through this term. Okay. This is an example. Yeah.
Sorry, I just had the question. What is the hyperfine interaction?
It's the coupling of an electronic spin with a nuclear spin. It's like what we have in hydrogen atom. In hydrogen atom, we talk about the hyperfine levels of the ground state that result from the electronic spin looking at the magnetic field produced by the nucleus. So this is the hyperfine coupling. So the two spins, nuclear and electronic, talk to each other because one produces a magnetic field sensed by the other, and vice versa. All right? So like if you have heard about the cesium clock used in time standards, the time standard that we still use is the so-called clock transition between two hyperfine levels of the cesium atom. Here we have hyperfine couplings in a biomolecule, but it's pretty much the same thing. So for example, here you can see by calculating the probability that the molecule is, say, in the triplet state, you can see nice oscillations that are due to this mixing between singlet and triplet radical pairs taking place. These are the green oscillations. If you open up the channel of recombination, the population of these guys is going down in time. Therefore, the green oscillation decays. And if you integrate that red curve over time, you show how much the total population in the products changes with time. At some point, the reaction is over, and this stops at the final yield, either in the triplet, here is the triplet yield, or in the singlet. So now, this calculation here is shown for one field. By changing the magnetic field, you see that you have a different sort of modulation between singlet and triplet, and at the end, a different yield in the products. So the magnetic field changes the hyperfine modulation of this oscillation and reflects itself in the actual distribution of products, singlet products versus triplet products. So by measuring those, you can learn what is the magnetic field. So we're talking about a biochemical magnetometer, right? as opposed to other magnetometers that people work on in quantum sensing technology. So we have worked a lot on addressing what is the master equation describing the dynamics of the system. You know, people in quantum optics work a lot with master equations. And we spent a lot of time to try to understand that because it's a rather complicated system. Of course, knowing the master equation is important because all observables are functions of the time-evolved density matrix, which follows these master equations. For example, reaction yields, reaction rates, NMR observables all follow from the master equation. So you'd better know it well. For a while, like since the 60s, people have been using a master equation that was empirically written down by Haberkorn, 1976, which involves the unitary evolution because of Hamiltonian interactions. And it had two terms involving recombination, so singlet recombination rate and triplet recombination rate. And these guys are projection operators that take the singlet part of the state and have it decay. So these terms reduce the normalization of the density matrix because, as I show next, the system is not only open in the quantum sense, it is also leaky. That means if you consider the radical ion pair, we have shown that the vibrations of the molecule form a reservoir that realizes a continuous quantum measurement of the spin state of the molecule. So there are two kinds of effects: real transitions that cause leakage of population towards the product states, but there are also virtual transitions that cause decoherence. So they disturb the continuous coherent oscillation of the spin state, and that causes decoherence. So the density matrix evolution has sort of two qualitatively different terms: real transitions describing a leaky system, and virtual transition describing an open system. So it's rather complicated. I don't want you to look at the math. Forget the math here. We have arrived at a master equation describing unitary dynamics from the Hamiltonian, dissipation of coherence. Actually, the Hamiltonian term generates coherence, and we're talking about singlet triplet coherence. This will come up again and again. We're talking about coherence between the singlet state and the triplet state of the molecule. So we have a generator of coherence, we have a dissipator of coherence, and we have reaction terms that reduce the population of the system, make it disappear. Plus, we might also have additional relaxation terms from the environment of the molecule. Okay? Now, these reaction terms involve a parameter, C of R, which quantifies coherence. And I'll come back to that theme because it is central. If we force this quantifier of coherence, C of R, if by hand we force it to be 0, we retrieve the earlier theory, which means that the earlier theory is useful in the regime of very strong relaxation, okay, very strong decoherence. So if the relaxation time is much shorter than the reaction time, the previous theory is fine. If it is similar or larger, so we have relaxation times much longer than the reaction time, the previous theory cannot work. In a nutshell, we have applied many tools of quantum information science to understand the system, like quantum measurement theory. The same reaction exhibits the quantum Zeno effect. We have used concepts from quantum entropy and information trajectories, and I will focus on quantum coherence quantification. One take-home message from all of that is that the coherence time of this reaction is similar to the reaction time. So it's comparable. The coherence lasts for as long as the reaction. This is not a non-trivial statement, so it needs all this study to understand why this is the case. We have also looked at Ramsey interferometer to realize a quantum metrological scheme measuring the magnetic field. That was a very simple, almost pedantic quantum circuit we used. But most recently, these authors from IBM actually performed a real quantum simulation of such reactions with IBM's quantum computer, and having real quantum circuits. And this is a very promising work for applying practical, currently, practical quantum computers, NISQ devices, to simulate such biochemical reactions. So coherence is central in this discussion, in particular singlet-triplet coherence. Now, the group of Peter Hoare has written this paper in 2020, how quantum is the radical pair magnetoreception. This is the central question. How quantum is it? And how do we quantify that? Now, in this paper, they use empirical arguments because at that point, there was no explicit quantifier for this kind of coherence. We introduced the first such quantifier in 2020 using quantum relative entropy. In fact, quantification of coherence had been introduced well before. This is a PRL by Plenio about quantifying coherence. So we adapted their metric to our case because we want singlet-triplet coherence. So we adapted the relative entropy definition by killing the singlet-triplet coherence and then comparing the entropy of this diagonal density matrix with the actual density matrix. So that's how we can quantify formally how singlet-triplet coherent is any given state of the radical pair described by Rho. Okay? So we have a formal quantifier of coherence, but then we did one more thing. Before getting into that, here we see how this actually works. It's rather obvious if we show a singlet-triplet oscillation plotted here on the left. So this is a probability that the system is in the singlet state, starting from 1, going to 0, now it's triplet, back to 1. This is a coherent oscillation driven by unitary dynamics. If you plot the coherence measure, you see that it is maximum in between. So when the system is in a superposition of singlet and triplet, that gives you the maximum coherence. That makes sense. Now, if you open the decohering dynamics, this oscillation decays in time. And of course, again, the coherence metric also decays. So it works out as expected. And it has all the proper properties. Now, we answered the second question is, OK, we have a quantifier of coherence. We can tell exactly and quantitatively how coherent is the state at any given time. Is there any quantum advantage? Because that's a basic question in quantum biology. Does the system gain anything in its workings due to the fact that it's coherent? What's the use of it? To discuss quantum advantage, we have to define a figure of merit. What does the system do which is biologically useful? Okay? If the system works as a compass, we can define, to simulate a compass, we define a Hamiltonian with a magnetic field rotating in some plane, xy plane, and the angle of the magnetic field in this plane is given by phi. And to realize a compass, one also needs an anisotropic hyperfine coupling, but forget that detail. So let's see how the compass looks. If we let this reaction run and calculate the reaction yield, that is the percentage of molecules ending up here, for example, and plot this yield as a function of this parameter phi, the magnetic field, we see a nice dependence, a nice oscillation versus phi. So the concentration of the reaction products depends on the angle of the magnetic field. Well, that's a compass. Okay, that's a compass. How good is this compass? Well, you have to see how sensitive this yield is to changes in the angle phi. The sensitivity is just the slope at some operating point. So here is our figure of merit. delta psi, delta y, is the slope of this line, defines how good a compass we have. All right? So now we have a figure of merit on the one hand, and we have coherence on the other. There you go. Here is the plot, the map of the figure of merit versus coherence. And to see this map, we included by hand various levels of decoherence, because that we can decide based on a little rate that we apply in decoherence terms. Okay, by increasing decoherence, you see that on average, the figure of merit of the system goes down. So this is, to my knowledge, the first quantitative explicit case that, yes, there is a quantum advantage in this biochemical reaction. Large coherence provides for a large figure of merit when we discuss the compass operation. Okay? So the figure of merit is sort of correlated with the singlet-triplet coherence C. And now I will discuss a more recent work, still in the archives, where we try to connect this reaction with physiological observables that are more readily measurable. So the title of this paper is Physiological Search for Quantum Biological Effects based on the Wigner-Yanase connection between coherence and uncertainty. Okay, so here's the idea behind that. First, we connect the reaction yields that we discussed with the reaction coherence, yield versus coherence. And then we connect the reaction yield uncertainty with further physiological observables. So thus, we have a connection between physiological observables through the uncertainty with the coherence of the reaction. So we were trying to look at a layer extending further beyond our core coherent reaction and see how coherence can manifest itself in physiologically relevant observables, because experimentally it could be easier to address coherence using, measurements in a little liquid sample, measurements involving concentrations and stuff like that. So one inspiration for this work was a paper in Nature dating in 2000, like 23 years ago, by Wiffer and co-workers, where they said the following. They implement a model that transduces the magnetic field dependence of the reaction yield. So, okay, we have the radical pair reaction. It starts with a photon. The radical pair is produced. It oscillates coherently between singlet and triplet. You get reaction products at these rates. And at the end, if you start with, say, 100 molecules in the singlet state, at the end of the reaction, you end up with a percentage here in the singlet and a percentage in the triplet, 20%, 80%, whatever. Okay? So the blue, dA, is your singlet yield. All right. So now they said, how is this yield informing follow-up processes in the cell? How does the cell know about this yield? Okay, so they said, let's build a little biological model involving cells that have membrane receptors. So that's actually quite useful in biology, where on cell membranes you have a protein that acts as a receptor. A receptor does what? It takes a ligand molecule floating around and binds to it. So here you have a receptor binding one of those ligands, which is actually this particular reaction product. All right? And then once the receptor binds a ligand, it transduces further changes in the cell. It's like how drugs work, basically. Okay, yeah.
And it's different from receptor and from the ligand.
Sorry?
And it's different from the type of receptor and ligand, what you get out of this or the measurements.
No, these receptors are supposed to bind only this kind of the singlet molecule, the blue. That's why I have the color coding. Okay, so receptors specialize in particular ligand molecules that they can bind. Okay. So suppose you have in a cellular environment cells floating around that have receptors that bind the singlet products of this reaction, and this binding is also described by another reaction, which is simple. It's like empty receptor plus molecule gives a bound receptor molecule. Simple enough. Now, how many such bound states you have is further transduced in the cellular environment into a signal, and finally, the end result is that if this changes too much, because the bird lost track, for example, the end result is some neuronal signal that tells to the bird, hey, turn, or some other physiological signal, whatever. But they built a model that uses receptor-ligand binding to further transduce the chemical reaction yield of radical pair. And they said the following. OK, I have singlet products plus receptor. In the steady state, I get a number of bound receptors called C. This has a given formula depending on this reaction. Don't mind the mathematics. I just want to give you the idea. And this is actually a classical discussion. There's nothing quantum here. If the concentration of these blue guys changes because the concentration L of those is proportional to the reaction yield that we discuss. So if the yield changes because the magnetic field changes, then that change will propagate into the number C of bound receptors. So we will have a change delta C due to the change of the concentration that was due to the change in the magnetic field. So finally, changing magnetic field, we have a changing number of bound receptors, which is further transduced biochemically, whatever the system wants to do. OK, so now they said the following. For the magnetic field change, delta B, to be measurable, the change in bound receptors, delta C, must be larger than some noise. OK, so they defined this noise, and this is a classical noise that is due to the probabilistic nature of this binding. So it's like Poisson fluctuation, simple classical noise. So based on this definition of measurability of the biochemical signal, they found that in order to have a magnetic sensitivity delta B minimum, that's the minimum measurable change in B, I need a minimum number of receptors to float in my cell given by this expression. So if I want this to be small, meaning very sensitive, I can measure a very small change in the magnetic field, I need a large number of receptors, which makes sense. All right? And dy over dB is the magnetic sensitivity of the radical pair reaction. So I need a sensitive reaction. So OK, that's classical discussion. So here's what we did. We inserted in this discussion quantum noise. How come? The reaction yield that comes into our discussion can be readily calculated if you know the density matrix of the system. We discussed that, forget, it's just an integral. But this yield is associated with an uncertainty because it's a quantum observable, after all. So we can calculate similarly, easily, it's straightforward, forget about the formula, we can calculate delta Y, which is the quantum uncertainty in the reaction yield. Now this translates into delta C tilde, which is a quantum uncertainty in the number of bound receptors stemming from a quantum uncertainty reaction yield. Okay? Now, this has not been discussed by those authors. So we entered this quantum noise into the discussion, and we said the following. Fundamentally, in quantum metrology, this is always the case, measurability means that the signal is larger or equal to the quantum noise. So in our case, the signal is this change in bound receptor number, delta big C. This must be larger than the quantum noise, delta C tilde. By requiring this, we retrieve the short noise limit on the magnetic field sensitivity, which we know from ordinary quantum metrology with atoms, photons, traditional quantum systems, right? we retrieve this shot noise limit, which is okay. We sort of knew that, nothing too surprising. But then we have another assumption. If the system is designed, quantum mechanically, if the physiological system has a quantum design, then the classical noise the authors use should be smaller than the quantum noise. Otherwise, the classical noise would dominate. And so that means you don't have a quantum design system, right? If the quantum noise, or if the classical noise is smaller than the quantum noise, then we arrive at another expression for the minimum number of receptors required to transduce the information on the magnetic field change. Now, the previous result of Wiffer and co-workers still holds, because the signal is still larger than the classical noise they had. We just inserted in between the quantum noise by requiring that we have a quantum design. Okay? So the connection between the minimum number of receptors and magnetic field sensitivity, delta B min, still holds as before. Only now we got two independent expressions for R minimum and delta B minimum, whereas before they were combined in one inseparable expression. So finally, here comes the Wigner-Yanase information. Wigner-Yanase information, of course, was defined by Wigner and Yanase back in the 60s. It's a common metric currently used rather frequently. So now we use the Wigner-Yanase information, which is of the density matrix rho with respect to some operator A, to define a new measure for singlet-triplet coherence. This is the Wigner-Yanase measure of singlet-triplet coherence, which is just I of rho and QS, the singlet projector, plus I of rho and QT, the triplet projector. So finally, we use an inequality discussed by these guys, Liu and Sun, in this PRF 2017. This inequality connects the Wigner-Yanase information with a variance of the operator A. So the Wigner-Yanase information is bounded by the variance of this operator. We use this inequality and finally arrive at a very interesting bound, that the time average coherence of the reaction is bounded above by the variance of the reaction yield. Now think about that. It's very interesting and non-trivial, because we connect the coherence in this step where the radical pair lives and oscillates with the variance of the reaction products. Okay? And this connection is, of course, an inequality, meaning that if we measure very small fluctuations in the reaction products, just concentration fluctuations of a molecule, that would tell us that the system is not very coherent. On the other hand, if we measure large variance, doesn't mean that the system is coherent, but it just allows us to look further. Okay, so connect time average coherence of reactants with the fluctuation of the reaction products. That's one thing. So now this is a formal inequality. We got before the minimum required receptors for under the premise of the quantum system design. So combining the two, we now have a new connection between the number of receptors required to transduce the information and the coherence of the system. So we connect biological observables, like number of membrane receptors, to the coherence measure of the system, of the core process. Okay? Of course, again, with an inequality. Okay? And the previous interpretation of how inequalities work, you know, it's an upper bound. So we connect the coherence of the core process up here, the radical pair reaction, with the number of receptors sitting inside a cellular environment floating around. This inequality came rather easily. A similar expression connecting the magnetic field sensitivity with coherence is not that easy to get. Therefore, we used a little simulation of an actual system, and we show that the magnetic field sensitivity is bounded below by an expression involving the coherence of the system. Again, like in the previous example with the compass, we show that singlet-triplet coherence seems to be a resource, because how sensitive the system is in terms of magnetic sensitivity depends on coherence. Large coherence means delta B is small, so you can measure smaller changes in the field. That's what you want. Okay. Ideally, this number should be as small as possible. So you observe that indeed, numerically you observe, that indeed coherence helps. Okay. So now we have two expressions, two inequalities, one for the number of receptors and one for the magnetic sensitivity. Now, finally, let's assume that these inequalities are saturated. So the system works in a parameter regime where these bounds are almost saturated. So you can multiply the two and get a very interesting expression, namely that the product of R minimum, the number of receptors, times the magnetic sensitivity square is equal to 1 over a power of the coherence measure. So now, if you look at that, it might remind you, like it did remind me, the uncertainty relation, only in a different kind of context, like a biological context. Why am I saying that? Because here we have the product of a biological resource, which is the number of membrane receptors. How many are there in the cell? Times a biological figure of merit, which is how sensitive the whole thing is, magnetic sensitivity. Now, we want both of these quantities to be small, ideally as small as possible. Okay? But they are not independent. They are constrained by the quantumness of the system, because C quantifies the quantumness. So, but this has the opposite meaning, the opposite dependence, like the usual uncertainty relation, where we have delta X times delta P equals quantumness. So the higher quantumness, H bar, the higher the product. Here we have the higher the quantumness. So the more coherent the system is, the smaller this product can be made. So the system can choose to operate with a small number of receptors transducing the information, or a small magnetic sensitivity, or a high magnetic sensitivity, or meaning delta B is small, but not independently, always so that the product satisfies this constraining relation. And that came again under the premise that these two inequalities, the former is formal, the latter is empirical, that these two inequalities are saturated. So I'm not saying that this is actually the case, but what I'm saying is that this can be the case. So this is allowed by the basic physics of the system, and it is possible that the system can operate in a regime where this is going on. So quantum design and the quantum trade-off between resources of biological, at the biological level, and the figure of merit of what the thing does, like magnetic sensing. Okay. So thank you for your attention, and I can close again with the same question that we started with, just to ponder and consider later on whether biologists need to learn quantum physics, or whether it's too early for that. In any case, thank you for your attention. Thanks to the funding program, and please feel free to ask me questions.
Also, we need to possibility of different orientation in the plane of the magnetic field, and then there was measurement of this orientation through the products that you have. But then there is not 2 pi, am I right? There is not 2 pi symmetry, because there are you can have the same products that corresponds to different magnetic fields.
Yeah, the whole thing is pi symmetric.
Yes,
Yeah, correct.
And I think this is also what they observed from phenomenological.
That's also observed by the biologists, exactly, phenomenologically. In fact, this is elaborated a lot in the papers of Vlitsko. The Vlitskos are a couple, an actual couple and a scientific couple that have worked, have done tons of experiments. Where is it? Here. This is one reference. They have written many papers on the Avian Compass.
Are they still active?
They are. I think I saw a recent paper a year ago. They are retired but still active, and they discuss what you correctly mentioned in their papers. Yeah.
Because then the birds from the north hemisphere, they fly towards the equator, but the birds from the south also to the equator.
Exactly.
And then this model.
Yeah, and then the model, of course, when you solve this model numerically, exactly. That's a very good observation. The model is pi symmetric. Yes.
Have they? I have another question. There is no in the meantime. Okay, I know for the models were there also on the basis of phenomenology, but have they really? Could they detect molecules that are magnetoreceptors?
Yes.
They could now, even biologically identify them.
Yeah. That has been done. I mean, this experiment by, I mentioned a couple of experiments. For example, you can look at the data here. This is the experiment of 2008 by the Hoare group in Oxford. And you can see here they measure the so-called magnetic field effect. And anyhow, this trace, you can see the change by, the change in the magnetic field is like from 39 to 49 microtesla. And they can clearly observe.
They are extracted from birds.
No, this is synthesized.
That's what my question.
Yes.
Could they see in the birds exactly, identify molecules?
They have identified the protein that carries these molecules, but I'm not sure if the actual molecules have been identified. Yeah. Yeah. Yeah, that's it comes out rather in a straightforward way, because let's go to the actual level structure here. So these are vibrational states that form a reservoir that actually performs a measurement of the spin state of the molecule. So there are two reservoirs because you have two reaction products, a singlet and a triplet. So if the initial state is the singlet, and we have asymmetric recombination rates, that is, if the singlet rate is much smaller than the triplet rate, then this reservoir also measures, this reservoir measures the triplet state, but it's the opposite as a singlet, because if you're triplet, you're not singlet. So essentially, if this measurement rate is strong, it projects continuously the state in the singlet state and does not let it evolve. So you can see that the oscillation between S and T is suppressed when the rate KT is very large. Huh? Yeah.
Yes,
Yannis, for a very stimulating talk. I mean, the second part was really find that in order to see a functional effect, there has to be some minimum scale of the photoreceptor.
Yeah, let me elaborate on that a little bit. So here we have, let's see. Okay, so far we were talking about the reaction of the radical pair resulting into products. But then again, if this happens in a solution, so the question is, if I cannot monitor directly the state of the radical pair up here, because I would have somehow to probe the state, the quantum state of the system in order to extract its coherence, if that's not easy, what else can I do to learn about coherence of this system? Well, the idea is that I can look at the reaction products by just looking at concentrations of the molecule. It's the typical measurement in biochemistry. So now, these molecules, the model says, will bind to membrane receptors, and the more of those we have, the blue molecules, the more bound receptors we have, and this information will be further transmitted to the system. Okay, so the connection we have made is the following, that we look at the fluctuation of this concentration, because this is a quantum observable, it has a quantum fluctuation. And to connect this fluctuation through Wigner-Yanase, we connect this fluctuation with the coherence up here. But the fluctuation of this molecule will translate into fluctuation in the number of bound receptors. So then by looking at the number of bound receptors or at the total number of receptors the system has chosen to have, which is another biochemically doable measurement, it's like measure receptors. Biologists can do that. We indirectly learn about the coherence of the system up here. So all this idea has to, as a goal, to connect quantities that were not apparently connectable, you know, like coherence with number of receptors. So you can have a solution, you can look at your cells, there are a number of cells in the system, you can measure the receptors, and this is a volume in which the cells are located, and this is a reaction constant, and see how this number that you measure is related to this bound, and from that extract information about coherence. Okay, I'm not saying that the information is one-to-one, because again, the bound, if the bound is if this number is small, you know, actually I'm confused myself. I mean, you cannot for sure claim that you have high coherence. Okay, you can tell whether you have small coherence, but if in the opposite case, you know that it is possible to have large coherence, but you can't prove that. You cannot prove that. So then you have a reason to look further.
Also, so it seems the key is your use of the Wigner-Yanase information.
Yeah, because
relying on a single operator, right? So you selected the Hyperfine operator there.
No, I select the.
Is it A?
Yeah, A is no. Yeah, forget about A. No. This is just some operator, but then I define a coherence measure, which this is actually formal. This measure of coherence includes the projectors QS and QT, the singlet and triplet projectors. We use them all the time in this discussion. So now, the Wigner-Yanase helps you define a formal measure of coherence. But the other advantage it has is that there is an inequality connecting the Wigner-Yanase information with the variance of this operator here. Okay? So that we transform into a relation between coherence and the variance of our reaction products. I think this is the cutest result. We connect fluctuations in some concentration with what is the coherence of the actual reaction. You know.
So
they can be anything in principle. In this model, these guys who proposed this model back in 2000, I'm not sure if they refer to any particular receptors. They just proposed this general model. Because if the reaction is actually being used in a cellular environment, it makes sense, that's my understanding, it makes sense that it will be transduced with this kind of receptor-ligand binding system, which is ubiquitous in biology. That's what I understand.
But it's really important that if they are from biological nature or if they are just synthetical, but if the paper is old, then I think that they use biological receptors.
No, they didn't. I don't think they specified whether they are natural or synthetic. But my guess would be that, you know, if one can study a natural system, one will find natural receptors. If you want to use synthesized systems, probably you would have to use synthesized receptors as well. I think the critical issue here is that these receptors only bind this guy and not this guy. Yeah. So they are specific for one type of molecule, which is actually the case in general, right?
I have a question related to what I was asking just a second ago. And sorry if you already explained this, but so this Y figure, the number of the yield of the thing, is I see it's integral over all time of something, right?
Yeah.
Like an equilibrium.
Yes.
Somehow.
Yes.
And so this delta Y is not a fluctuation in time. It's not like
No, this is, yeah, no. It's like if you repeat the experiment, you will get a distribution of y's that has a given width.
and this, okay, so basically to repeat the experiment, I have some sort of, I have that system sitting there for a long time.
Yeah, you have to, you need to insert a time scale because this is supposed to flow. So you have a compartment where this takes place, and then these molecules flow into another compartment, and there's a continuous flow. So you know you need a time scale for all that. So.
Is delta Y is.
So the delta Y is the delta Y is if nothing changes here, if all the parameters are the same, if you repeat the experiment, the Y you will get will not be the same, but it will be distributed around the mean, the mean Y with a given width given by delta Y.
And the noise is proportional to the coherence. It grows with the coherence of the reaction of this HB in the middle.
Yes. So yeah. So yeah. So if you have very little fluctuations, it means that you have little coherence, because it makes sense, right? Because if you have superpositions of things, you have a large variance, right? If you are only in one eigenstate, you have little fluctuations, and there is no coherence because you are sitting in this eigenstate, right? So this makes sense physically. And again, let me stress that if this is small, then you learn something that is sort of negative. Oh, well, coherence is small. You don't like that. You usually like large coherence, you know. So you get a negative information, I mean, coherence small. But if this is large, it doesn't mean that this is large, right? Because yeah, exactly. Could be completely mixed state. Yes, exactly. Yeah.
Okay, so thanks once more.
Thank you. Thank you.